Hello. Okay. So today we are going to start our session on our pathology, right? So the first topic which we are going to discuss is our neural tube defect, right? So the first topic which we'll be discussing is neural tube defects. But before discussing neural tube defect, as we guys already know the normal embryology, that we have our ectoderm, we have our mesoderm, and from the mesoderm a structure would arise which is known as our notochord. If you guys remember, in our mature individual we have remnant of notochord as well. If you guys remember nucleus pulposus, which we discussed, right? So this notochord would just keep on sending signals to this ectoderm, and ectoderm would turn, it would just transform into a structure which is known as neural plate. And this neural plate is important for making all of your nervous system different cells, right? So this neural plate is going to invaginate and it would just form a neural tube like that. But remember, at the sides of your neural tube you are going to have specific cells which are known as neural crest cells. Most of you guys are already aware of neural crest cells, that it contribute in formation of your peripheral nervous system. It also continued in formation of your Schwann cells. You guys are already aware, the medulla of your adrenal cortex, right? So once you have complete formation of this neural tube, so from the caudal, or from the cranial to caudal aspect, you have pores within this neural tube, right? And those pores have to be closed in a mature individual. If we have failure of closure of these neural tubes, we are going to end into a certain condition which we are going to discuss, right? So for example, you can think of it like a tube. So you have a anterior neuropore and posterior neuropore, right? So an adult individual, you need these pores should be closed, right? If the anterior pore do not close, it would end into a condition which is known as anencephaly, right? So we are going to have a lot of UL MCQ on that. What is anencephaly? Basically, different parts of your brains are going to be missing. We are going to discuss that, right? And if we have failure of closure of posterior neuropore, it would end into condition which are known as spina bifida, right? And we have a lot of types of spina bifida which we'll be discussing, right? So an important point which you guys must keep in mind is that for the closure of these neuropores you require folate, right? Before conception. So before conceiving, a mother should have an adequate intake of folate, right? But another point which you have to mention, that there are a lot of drugs which basically interfere in metabolism of folate. Folate, as we already have discussed in our anemias lecture, it's important for build up of your DNA, right? We already discussed that. So folate is very important. So certain drugs which are for example methotrexate, you guys are already aware of, they basically decreases they're anti-folate simply, right? So it can cause problems, for example the inability to close anterior neuropore or posterior neuropore, and this child would end in anencephaly or spina bifida, right? Similarly, another drugs, for example valproate, which is basically used in a bipolar disorder, it can also interferes the folate metabolism, and in the end it can lead to anterior or posterior neuropore defects, right? So you have a UL MCQ on that, remember, right? So you are going to have a lady who have bipolar disorder, and she just she's using certain drug. For example, what is that drug, right? So you guys should know the valproate, right? Okay, so whenever we have anterior neuropore defect or posterior neuropore defect, okay, these are simple pores which have to be closed, okay? So it's going to give you a lot of other things as well. You should just keep in mind. For example, look, the idea is very clear. If the anterior neuropore do not close, or either the posterior, it means that all of the components of this embryo is going to leak into your amniotic fluid. Amniotic fluid we already discussed. It's basically fetal urine, right? So what things are we going to see which is going to indicate inside a uterus that a baby is having either anterior neuropore closure defect or posterior neuropore closure defect, right? So remember two things: alpha fetoprotein, right? Alpha fetoprotein, and you should just simply think of that Alpha fetoprotein, it's a part of a fetus, and if these pores do not close, it would just leak into the amniotic fluid and AFP level would be increased. And another thing which you guys should just keep in mind is acetylcholine, right? Acetylcholine. So you guys should just keep that in mind, right? Why acetylcholine? Remember that the acetylcholine basically, it is concentrated in your CSF, right? So in the CSF you have a lot of acetylcholine. So if you have increased acetylcholine within your amniotic fluid, so it basically suggests that the CSF of this child is leaking into your amniotic fluid, and you can just suspect it for closure defect, right? Anterior or posterior neuropore closure defect, right? Okay, so let's discuss our anterior neuropore further. Anterior neuropore defect. As we already discussed, whenever we have problems with anterior neuropore, what happens? It would lead to a condition which is known as anencephaly, or in other words, the different parts of the brain are going to be missing, either the skull or different hemispheres, right? So you have a UWorld MCQ on that. It's going to give you a presentation of a child who haven't developed his skull or different parts of the brain. So what's the condition going on there? That is anencephaly, right? And another point which you guys should just keep in mind, we know that for the normal amniotic fluid cycle, right? Amniotic fluid, we already discussed, it's a fetal protein. It means it's made by the kidney, and for the cycle to complete, the baby have to swallow it, right? The baby have to swallow it. So in this way you are going to have a constant amount of amniotic fluid. But if we have anterior neuropore defect or anencephaly, it also leads to inappropriate formation of mouth as well. So if the mouth formation is inappropriate, the idea is really clear. Now the baby cannot swallow your amniotic fluid. If it cannot swallow, it would end in a condition which is known as polyhydramnios, right? Polyhydramnios, or a lot of amniotic fluid. Is that clear? Okay. So this was all about anterior neuropore. For example, if the posterior neuropore do not closes, it would end in a condition that is known as spina bifida. So what happens in spina bifida is that the posterior vertebral arches do not close. Like this posterior neuropore is important in order to have a proper vertebral arch. If the vertebral arch do not close, the idea is really simple. If you do not have vertebra, all of the content of the CNS can herniate through the spore. The idea is really simple. So we have a lot of variants of spina bifida, right? So the first variant of spina bifida is called as spina bifida occulta. So what happens in spina bifida occulta is that you are going to have a dimple or a patch of hair over the defect, right? So whenever you see a patch of hair over the defect, and else it's like, I would say, like least severe form of your spina bifida, right? So only you are going to have a tuft of hair with a dimple, which indicates failure of closure of posterior neuropore, right? Okay. And the other variant are spina bifida, meningeocele, and meningomyelocele. The idea is really clear. Meningeocele, like sometimes only the meninges can herniate through that small opening due to inappropriate closure. So that would be meningeocele. And the word meningomyelocele means myelo refers to your spinal cord, right? So the word meningomyelocele refers that in this variant of spina bifida, you are going to have herniation of both your meninges along with your spinal cord, which is the severe form of your spina bifida, right? So it was all about your neural tube defects, right? So the other topic which we are going to discuss is cerebral aqueduct stenosis. Cerebral aqueduct stenosis. As we guys are already aware of the normal ventricular system, that we have our lateral ventricle. Lateral ventricle would just join your third ventricle via foramen of Monro. Third ventricle is going to move to your fourth ventricles through your midbrain via cerebral aqueduct, right? So sometimes what happens, this cerebral aqueduct can get stenosed. If the cerebral aqueduct gets stenosed, the idea is really clear. We already discussed choroid plexus basically makes CSF. So So that if you are just blocking the pathway for the CSF, what happens? All of the CSF is going to build up and it's going to result in hydrocephalus, right? So what would be the presenting features? A baby with cerebral aqueduct stenosis would be having increased, yes, head circumference, right, due to accumulation of all of those CSF content, right? And similarly, it can also like cause, for example, if we have closure of the Lushkas and Magendie, which we have already discussed, it can also lead to, yes, your hydrocephalus. Okay, so next topic which we are, okay, we have a UN MCQ, if you guys remember, that is pineal gland, right? So pineal gland, also known as third eye, right, is responsible for secretion of your melatonin. Sometimes this pineal gland can also get, like hypertrophied, or it can also come and it can also, like block of this pathway, or in other words, it can also cause cerebral aqueduct stenosis and will be having the same features as we discussed, right? So you have a UL MCQ on that as well, right? Okay, so the next pathology which we are going to discuss is Dandy-Walker malformation. Dandy-Walker malformation. What happens in Dandy-Walker malformation is that we are already aware of the structure of cerebellum. If you guys remember, so we have cerebellar hemispheres. In the middle we have an area which is known as vermis. Surrounding the vermis we have paravermis, and below we have a flocculo-nodular lobe, which is important for balance of your, yes, which is important for your balance purposes, right? Okay, so the vermis, as you guys already know, the homunculus present on the vermis is basically responsible for your axial skeleton, right? Muscles of your axial skeleton, while on the paravermis you have homunculus for your extremities, right? So what happens in Dandy-Walker malformation, you guys are already aware of. For example, if this is your, okay, or let me just draw the structure of your brain. For example, if this is your brain, okay, so we have our pons, we have our medulla, and we have our spinal cord. Then we have our, okay, so here you guys can see we have our cerebellum at the back, and this is, let's suppose, the vermis part of your cerebellum. And we are already aware that the fourth ventricle sits half in the pons and half in the medulla, right? So what happens in Dandy-Walker malformation is that the cerebellar vermis do not develop, right? So it's a congenital failure of cerebellar vermis to develop. If the vermis do not develop, there are going to be a lot of problems. For example, cerebellar ataxia, right? And if the vermis do not develop, it can also compromise the fourth ventricle, right? So the fourth ventricles, as we know, it is present in close proximity with the cerebellum. So what happens? The fourth ventricle is also compromised, the formation, right? And if we have compromise of the fourth ventricle formation in the posterior fossa, again it would lead to closure of these foramens, Lushka and Magendie, which can in turn again cause hydrocephalus, right? So you have a UL MCQ on that. They will be just giving you a clear picture of dilated fourth ventricle, which suggests hydrocephalus, with improper formation of vermis, which is going to cause problem. For example, cerebellar problem. Ataxia is specifically, we know, the homunculus of the axial skeleton. Is that clear? Okay. So the next topic which we are going to discuss is Arnold-Chiari malformation. Arnold-Chiari malformation. So what happens in Arnold-Chiari malformation? I would say it's a less like harmful variant of the displacement of your cerebellum, right? So sometimes what happens that, for example, you know that this is your foramen magnum and you have your brain here, right? You have your pons and the medulla, and here you lies the cerebellum in your posterior fossa, right? So sometimes what happens, some of the component, that is the tonsils of cerebellum, can herniate down to the foramen magnum. So this is your foramen magnum, right? And once it's herniated down to the foramen magnum, it can cause problem, right? Okay. So we have two variants of Arnold-Chiari as well, Arnold-Chiari 1 and Arnold-Chiari 2. 2 is like, I would say, like a more severe form of Arnold-Chiari, right? So whenever we have herniation of cerebellar tonsils to the foramen, that's Arnold-Chiari 1. But if the cerebellar vermis and the tonsil both just herniate, okay, this is the vermis part, if both herniates down through the foramen magnum, that is simply your Arnold-Chiari malformation too, and it can cause a whole lot of problems. For example, if all of the brain keeps on herniating through the foramen magnum, like for example the tonsils, the vermis, the brain stem will also keep on herniating, which can cause a lot of problems, because we have a lot of centers within the brain stem, within the medulla, right? We have respiratory centers, there's a lot of things there, right? Okay. An important association of Arnold-Chiari malformation too is that it's highly associated with meningomyelocele, right? Meningomyelocele. And what was meningomyelocele? We already discussed. It was defect of posterior neuropore, in which we are going to have herniation of both your meninges along with your spinal cord. Is that clear? And the idea is very simple. If things keep on herniating back, again it would cause hydrocephalus, because the pathway of the fourth ventricle is disrupted here, right? Okay, so the next topic which we are going to discuss is spinal cord lesion, right? So as we have already discussed spinal cord in detail, we already know the tracks, we know everything. So as I already told you guys that if you guys know the anatomy, every pathology is going to be really easy for you guys, right? So we have spinal cord lesion, right? Before discussing spinal cord lesion, let's discuss the anatomy which we discussed. We discussed that we have our spinal cord, in between lies our beautiful butterfly, right? And we have a spinal canal here. We discussed that here you will be having dorsal column, medial lemniscal system, in which the modern sensations, for example, your vibration, 2 points discrimination, proprioception is going to be carried through fasciculus gracilis, cuneatus, if you guys remember, right? And in the lateral section we discussed, we have cerebellar fibers, the anterior spinocerebellar, the posterior spinocerebellar. Along with that, we also have the descending fibers, corticospinal fibers, which will just get it will just get signals to your anterior spinal cord, which is anterior horn, which is motor in nature, right? We also discussed that we have lateral spinothalamic tract, which would just carry information, that is your pain and temperature. If you guys remember what happens in the lateral spinal cord tract, that things would just come through the posterior horn, and it would just relay here in the dorsal horn, and it would just move 1 or 2 segment above, and it would just come through the anterior commissure like that, and then it would just start moving in the lateral column up to your brain. We already know that, but this part you guys should know that 1 to 2 segment above in anterior commissure, right? So what happens in our first pathology which we are going to discuss is syringomyelia, right? Syringomyelia. What happens in syringomyelia is that we are going to see formation of a syrinx. What is syrinx? Syrinx is simply cystic degeneration of your we have cystic outgrowth from the spinal canal, which would just come in front, and it would compromise your anterior commissure, right? If this syrinx is compromising your anterior commissure, the idea is very simple. We are going to have loss of pain and temperature where 1 or 2 segments below the lesion, if you guys know, right? Because all of these pain and temperature fibers are going to move 1 or 2 segments above in your anterior commissure, right? So this was all about syringomyelia. But later on, what can happen, that it would not only affect the fibers of your anterior commissure, but it can also involve other spinal tracts as well. For example, with time it can involve your anterior horn, right? And we already know that in the anterior horn, basically lower motor neurons are present. So if we are knocking off the lower motor neuron, we are going to see signs and symptoms of lower motor neuron lesions. And what are the signs and symptoms? We are going to see fasciculations, we are going to see fasciculations, we are going to see low atrophy, we are going to see hyporeflexia, right? Hyporeflexia. So all of these symptoms are basically associated with your lower motor neuron lesion. And sometimes what happens, this syrinx can also involve the lateral horns as well. If you guys remember, lateral horns have neurons of your sympathetic nervous system, right? So if we have lateral horns being affected, it would just if the sympathetic is knocked down, we know it's going to end in a condition which is known as Horner's syndrome, stenosis, meiosis, and anhydrosis on ipsilateral side, right? So three things to keep in mind. Syringomyelia. Syrinx can invade three things, right? Initially, it would invade your anterior commissure. As we know that in the anterior commissure, fibers of your pain and temperature are going to cross, they are going to move, right? So you are going to have deficit of pain and temperature. It can invade, it can cause degeneration of your anterior horn as well, which would lead into, yes, it would lead into lower motor neuron symptoms, right? Because lower motor neurons are present in your anterior horn. It can also compromise your lateral horns, leading into Horner syndrome, disruption of your sympathetic nerve fibers, right? Okay. So the next pathology which we are going to discuss is poliomyelitis. You guys are already aware of this virus, poliomyelitis. It's a poliovirus. It's basically transmitted through fecal-oral route. And remember, hematogenously it can spread into your central nervous system. Specifically, it would invade and it would knock down neurons in your anterior horn. And we are already aware of that. If the anterior horn lower motor neurons are knocked off, again it would lead to same fasciculation, atrophy, and hyporeflexia, right? So like you guys only have to keep in mind different locations in different pathology, right? Like in syringomyelia, anterior commissure, you guys already know that, right? Pain and temperature crosses there. Anterior motor neurons, lower motor neuron signs and symptoms. Lateral horn, which have sympathetic neurons there, right? So this was about poliomyelitis. You also have a same kind of variant, same as that of poliomyelitis, which is known as Werning-Hoffmann disease. But the only difference is that poliomyelitis is due to virus, while Werning-Hoffmann is an inherited, okay, let me just write down the name, Werning-Hoffmann disease. What happens, like similarly, each and every signs and symptoms and the location of the lesion is same as that of polio, but the only difference lies in the cause. Polio is due to virus, while it's an inherited defect in which we are going to see degeneration of your anterior horn lower motor neurons. If anterior horn lower motor neurons are knocked off, you guys already know that what will happen. It would end in fasciculation, hyporeflexia, and lower motor neuron signs and symptoms. Is that clear? So this was all about Werning-Hoffmann disease. Okay. So the next pathology which we are going to discuss is amyotrophic lateral sclerosis. Okay. What happens in amyotrophic lateral sclerosis? Again, it's a pathology which results in degeneration of upper and lower motor neurons. Remember, both upper motor neurons and lower motor neurons are affected, right? So you're going to have like a kind of both signs and symptoms of upper motor neuron lesion and lower motor neuron lesion, right? So remember that what happens that, let me just draw the structure again. For example, this is our spinal cord, our beautiful butterfly, right? So we have, okay, so what would happen in amyotrophic lateral sclerosis? It would lead to, like this condition would lead to anterior motor horn degeneration. In the anterior motor horn degeneration, we are going to see lower motor neuron symptoms. We already know that, right? Lower motor neuron symptoms, because all of the lower motor neurons are present within your anterior horn, which we have already discussed, right? We will be having muscle atrophy, fasciculations, and what will happen to